Hi everyone, my name is Bharat Raj and I am studying in FI Biotech from Pillai University in Navi Mumbai, Panvel, Maharashtra. So today I am going to presenting a small detail about DNA replication, the foundation of life, how it's work, how it's going, everything I will tell you in this video. So be ready with this. DNA replication is the biological process by which a cell make an exact copy of DNA before cell division. So it is known as DNA replication. I give a small definition of a DNA replication and their process. So DNA replication is a process in which one molecule produce two identical DNA molecule. That genetic information is passed accurately from parent cell to daughter cell. So it is the small definition of DNA replication. Why DNA replication is matter? So there are two main types. So first type is cell division and their growth, and the another one is genetic inheritance. In cell division and their growth, every time cell divide, it must create a perfect copy of DNA. The process enable organism to grow, repair, damage their cells and tissues, and replace old cells with new cell throughout their lifetime. So it is in a cell division and their growth. Initiation finding the starting point, reconjunction of origin. So its replication began at specific sequence called of origin replication, where indicator protein recognize and bind to 80 reach recognize of DNA, and protein complex exemplarily multiplied rather to originate site, forming pre-replication complex that prepare the DNA of unwinding and copying of the replication. And fork formation in a helices loaded to a DNA beginning to unwind the double helix and creating two replication fork that move in a opposite direction. Bond between complementary base pair. The enzymes the enzymes use energy from ATP. It's it's utilized to power in the moment, advancing at a rate of approximately 1000 base per second in a prokaryotic. Stabilizing and subtracted stand. So it has stabilized, and so as a helicase unwind the DNA single strand bonding bonding protein SSB immediately caught the cost single bond. These proteins prevent the which prevent the strand from arising or form any secondary structure that will interference with replication. It is mainly provide us bonded strands, helicase action and stabilization. So leading start continuous synthesis. DNA polymer synthesized continually in the same direction as the replication of a frog. Moving requires DNA polymer to start the follows naturally direction of a DNA polymer, making replication straightforward and efficient. So in a lagging standard discontinuous synthesis, DNA polymers must work away from the fork direction, synthesizing in the fork segment called Okazaki fragment. Each requires its own DNA primer, RNA primer. So this is created as a complex processing requiring the coordinator of multiple enzymes to create a complete standard. Polymer synthesis. RNA polymers set the stage. The premier requirement, DNA polymers cannot be synthesized from starch. It requires a pre-executing 3 OH group to nucleate this where premiers enter the picture. So in a premiers create DNA premiers is a specialized for RNA polymer synthesis. Short RNA specific over 8 to 12 nucleotides that provide the unnecessary 3 OH group of DNA polymerase to begin continuous versus repeated premier. It leading stand required just one premier origin the lagging strand and need to premiere 1000 to 2000 nuclei to initiate each Okazaki fragment. DNA polymer in action adding nucleate with precision. Template leading. So DNA polymer read the template strand, identify each base A, T, and C or C. Determine which complementary nucleide should be added next. Nucleide selection. So that in a nucleide system, the enzyme start the correct free nucleide from the cellular pool following base pairing rules. Adenine pairs with to collect the pairs with cytose, so it is known as cytone. So and the postproteases bond DNA polymers catalyze the formation of a covalent bond between the 3 OH group of forming strand and the 5 phosphate of the in incoming nucleide. So advancement, after adding each nucleide, the enzymes move the position along the template, repeating the process with extraordinary speed and accuracy. So there are, you can see the speed will be 1k nucleide added per second, and the accuracy 99.99% is the accuracy base pairing. So in this video we talk about DNA replication, the foundation of life. So hope so you like the video. Thank you.